Okay, students, coming to an end of the session, we've got our question number 14. And question 14 is again, describe the following. Okay, so what we are supposed to do is, we're just supposed to describe what these four terminologies are. Okay, so transcription. Transcription is formation of RNA over the template of DNA. So transcription is mainly about formation of RNA from DNA. It forms single-stranded RNA which has a coded information similar to the coding strand of DNA with an exception of thymine. So instead of thymine, we will have uracil is replaced by uracil. One of the strand of DNA is used as a template and the other one is formed around it, which is known as complementary strand of RNA called mRNA. So students, this is what is transcription. Now, going ahead with polymorphism. Polymorphism. Poly means more. Therefore, this form of polymorphism, in this form of polymorphism, there is variation in DNA at a given genomic site. So at one site, DNA would be different amongst individual of a population. Now, it is a tree, for example, SNP, that is single nucleotide polymorphism. It can be of three types. One is single nucleotide polymorphism. Second is VNTRS, that is variable number of tandem repeats. And RFLM, that is restriction fragment length polymorphism. Polymorphism means change at a specific site of a genome. Okay, and it can be of three types. One, that is single. The other one is variable, and third is restriction fragment length polymorphism. Now we are done with these two. Let's start with these two. Translation. Okay, translation is a process during which the genetic information which is stored in the sequence of nucleotides in an mRNA molecule is translated. What? It is translated following dictation of genetic code into the sequence of an amino acid in the polypeptide. It occurs in cytoplasm in both eukaryotes as well as prokaryotes. So, in translation, whatever the sequence is stored, whatever the information is stored inside our genetic material is translated. Okay, so that is what is translation. Now, lastly, bioinformatics. So, it is a combination of biology and IT. Biology, IT, and computer science that deals with the data storage, retrieval, and analysis of sequences based in human genome using high-speed computational devices. So, it is a mixture of what, students? It is a mixture of information technology, science, that is biology, and lastly, computer sciences. It will analyze, it will read, it will be retrieval of data, and so on. Okay, students? This science is useful in organizational biological data, functional genomics, and proteomics in diagnosis and treatment of certain diseases. So it's one of the most leading branch of biology, and students, it is an amalgamation of IT and biology all together. So with this, we come to an end of the session. Okay.